Hi, everyone, and welcome to our live adult-focused neuromuscular seminar today. I'm Nicole Petrowski. I am the Community Education Specialist here at MDA, and I wanted to thank also our supporter for today, Amicus Therapeutics, and also any of their representatives who are on the line who might be listening today. Here at MDA, we're committed to community education, and we believe in the power of bringing our community together for opportunities to learn from specialists and have opportunities to connect with others. This event is part of our larger MDA Engage series with community education seminars, which we are having now, disease-specific symposium, and community webinars as well. Here is a look at our upcoming community education seminars we are going to be hosting for the rest of the year. These are day-long educational seminars which provide individuals and families with resources around neuromuscular disease and related topics such as care and research and resources in these general areas. And this is a schedule of our upcoming disease-specific symposias. These are also day-long seminars, but they are geared only to those diseases that we're highlighting. So everything is related to that particular disease. And then finally, here are our upcoming community webinars, and these are a little bit different. They're only an hour, and each webinar will feature experts in the field, and also you will have opportunities to ask questions along the way. And before we begin, I have just a few housekeeping items to go over, and you can see we have a pretty large packed day here. So we are recording today's event, and we will be posting it to our mda.org website for on-demand viewing in a few weeks. For those of you who are joining the event live, please know all phone lines have been muted. And we will be having Q&A session at the end of each presentation. So if you can please type in your question in the Q&A icon at the bottom of your screen and enter it to host, that would be wonderful. And you don't have to wait till the end of the presentation to submit your questions. And with that, I would like to introduce our next speaker. Dr. Mazen Dimachkie is a professor of neurology, director of neuromuscular division at KUMC, and director of the Neuromuscular Medicine Fellowship. He is also executive vice chairman of the neurology department and vice chairman for research programs. Dr. Dimachkie manages one of the busiest neuromuscular clinical trials units in North America, which includes federally funded neuromuscular research studies and industry-sponsored studies nationally and internationally. He is a frequently invited lecturer on inflammatory muscle disorders, Pompe Disease, Myasthenia Gravis, LEMS, ALS, neuropathies, and other neuromuscular disorders. And he has more than 200 publications, 300 meeting abstracts, and 29 book chapters. So thank you so much for being with us today, Dr. Dimachkie. You're very busy, so I definitely appreciate you taking time to be with us. And I will turn it over to you, and you can show us your presentation.
Thank you, Nicole, for this very kind introduction and for the invitation for MDA to invite me to engage with patients. I see we have more than 100 participants. I don't think they're joining because of me, but because of the rest of the great program that you set up together as part of this outreach program to patients. Of course, when I see patients in my clinic, the question that comes to mind is about what's new to help my health be better. So hopefully today, while I cannot cover all the advances because they're numerous, I'll focus on a few disease states that have to do with muscular dystrophy, specifically 4 of them, as we move forward. Of course, as Nicole mentioned, put your questions, please, in the chat box. I don't have numerous slides. It's only about 33 slides. I'm not going to run through them. We're going to probably need about an hour or less to go over those slides, but we'll have plenty of time for the Q&A session. So thank you again for joining me today. One of my passions is patient education, and so I always look forward to hearing from patients also about their perspective about how well we're doing, what are we missing. So thank you for your candid input in advance. Having been in the research and education field for quite a while, I have several disclosures listed here. Some of the research that I'll describe to you I may have been a consultant on or participated as a site in or received educational grants from companies that support that research. And none of those, potential conflicts of interest will get in the way of me giving you an objective description of what's going on in the landscape of neuromuscular disease with the focus on those four diseases. So when I teach students, residents, fellows, or experts in international conferences, we start with what's called learning objectives. And please indulge me here. But of course, what you see here on the right side is the picture of someone that's really special, that coined the name, his name to a disease that we're going to talk about, ALS. That's Lou Gehrig. And we're going to be first starting by reviewing the different types of research studies, because some of us, um, you know, um, when we think about research at the layperson level, I'm going to make an assumption, and excuse me if I'm making the wrong assumption, but I'm going to assume that all of us wondering, why is it that there's a drug that's promising and it takes so long to develop it? What are the stages of research that it goes through? And how can it be accelerated? And what types of research are there out there? So I'm going to try to go in generic terms, but with some specificity about the types of research studies that are out there and the stages it goes through. The early stage, the mid stage, and the late stage, we call them phase 1, phase 2, and phase 3. Some of you may know this, but maybe not all of you. Then I was going to go over what we know currently about standard of care management for diseases like ALS, amyotrophic lateral sclerosis, also known as motor neuron disease, also known as Lou Gehrig disease. Then I'll be discussing myasthenia gravis, an autoimmune disorder of the neuromuscular junction, then dermatomyositis, an autoimmune disorder of the muscle itself. And you can tell my bias is a little bit towards the muscle, because I'll be talking also about Pompe disease. Now, when I decided on those topics, I did not know Amicus is a supporter of this program. I would just want you to know that. And I was a participating PI in their Amicus study, as well as other Pompe studies. But again, nonetheless, I'll present to you the data as I know it and the information as I know it. So what I'll do, besides talking about where we're at now, is where we're going next, because we need to know what are the promising research studies in these four diseases. And I wish I could tell you about all neuromuscular diseases, but as Nicole mentioned, there's so many advances, and it's really hard in the limited time to talk about all of them. We would need a full day, 8 hours, maybe, to scratch the surface. So next, I'm going to discuss the types of research studies that you can help in, because we need your help when it comes to research. We can have a great idea and great plans for doing this test in the research and having the right research personnel, but if we don't have the patient engagement part of it and contributing to the research, we fail. So the simplest form of research is you come to the clinic, and as part of your clinic visit, you agree to have your data entered that's obtained in the clinic in a database. And that's called registries. They don't change what is done in the clinic. You get the regular standard of care, but the data is abstracted, is taken from the chart with your permission as part of a registry. The other type of study that's listed here on the second bullet point is something called natural history. And I should have put natural in quotation mark, because once you put someone in a natural history study, what you're doing is every 6 months to a year, or maybe every 3 months, depending on the nature of the disease, you'll bring them in for a clinic visit whereby you have specific instruments, outcome measures, assessments, tests that you run on them periodically. So it's kind of natural, but not so natural, because that's not what happened in real life, but that's the closest to a natural history that we have. Then the idea behind those registries and natural histories, especially the natural histories, is that the best way to do research on a disease is to know what is the best measuring stick. What's the best measuring stick? Because if you know a disease, how it behaves over time, and you know how the measuring stick evolves over time, then if you have a treatment, now you have a good measuring stick to measure the effect of the treatment. There's sometimes other forms of studies, genetic studies for diseases, looking at modifiers of the disease expression. Not everybody with, for example, Duchenne muscular dystrophy gets weak exactly the same way. Looking at gene modifiers, same for Pompe, same for other genetic diseases. Even in acquired immune diseases, there could be modifiers there that are genetically based, in addition to the environment, of course. Biomarkers, for example, it could be a blood biomarker or it could be an MRI scan biomarker. If you have muscle weakness from muscle disease, does it make sense to measure the thickness of the muscle, how much fat or how little fat infiltration there is in the muscle, how much inflammation or water uptake there is in the muscle? So these can be, if we know the natural history of those, and we have an intervention that changes that trajectory by a certain percent, that gives us an idea we're on the right track for success. Now, beyond these preparatory studies, next come the next level of preparation. Many of our diseases, but not all, have animal models to support them, meaning a model in a rat or a mouse or a monkey that looks like what happens in human, whereby you can take a drug and test it. These are called preclinical studies. So these are the preclinical. The clinical trials are in phases, phase 1, phase 2, 3, and phase 4 is when the drug is on the market and you're doing a post-marketing surveillance. That's typically done by a drug company, and it's usually an FDA requirement. But phase 1 is the earliest phase. This is where we're looking to make sure that the drug doesn't hurt you. And that takes time to do. Then phase 2 is more so finding the right dosage of the medication or trying to do a placebo-controlled study in a small number of patients, 20, 40 patients, small number. Whereas phase 3, this is when you have about 100 or more patients at multiple sites enrolled, and now you found the dose that's right from phase 2, and you're trying to apply it across the board. And this is the reason why it takes such a long time to develop drug. First, you have to have this data about what's the best measuring stick. You also have to have some preclinical information about the efficacy of the drug, or at least some phase 1 study that is safe and there's some signal. And then you move through the phases. So it's not unusual for drug development to take 10, 15 years from starting in the early phase to get to the answer if it works or not. Now, when I talk about research studies, most people get excited about these last 2 bullet points: stem cell therapy and gene therapy. Gene therapy is kind of like the holy grail. We want gene therapy for those diseases that are amenable to gene therapy, but not all of them are. We know that. But stem cell therapy, it depends also on what organ is involved when it comes to the stem cell therapy. The strategy may be quite different. So when we think gene therapy, about 20 years ago, when there was decoding of the human genome, there was a lot of excitement that gene therapies are now going to be available within the next year or two. And then, of course, time passed, and gene therapies did not come on the market or become available. But now it is available. There's several different forms of gene therapy currently available in the field of neuromuscular disease and in the muscular dystrophies. For example, on the market, earlier, skipping of exon 45 was mentioned by Nicole, but on the market already, we have Exondys 51, which is a drug for Duchenne muscular dystrophy to skip this exon and improve and stabilize patients with Duchenne muscular dystrophy. There are some treatments where It's not skipping a exon. Exon is part of the gene that's expressed, I should have clarified, whereas intron is usually a non-expressed sequence of the gene, but can have an effect to modulate by splice areas the expression of the gene. So the other approach is if you can package the gene or a critical portion of it, or modify the gene somehow and use a vector, and the vector is typically a adeno-associated virus or lentivirus. That's what AAV stands for, LLV stands for. So you take a shuttle that you inject into, typically intravenously, and in that shuttle, which is the AAV virus or the LV virus, is a gene for a recessive disease like spinal muscular atrophy. And then depending on how big the cargo capacity of the shuttle and the type of carrier, as well as the immunogenicity of the virus, because we are exposed to these viruses. Are we immune to them? We get a body immune reaction. And what reactions happens outside, say, the muscle? If this is your target organ, what's happening in the brain? What's happening in the heart, in the kidney, in the liver? Are there some damages occurring as a secondary bystander effect? But clearly, there's success in spinal muscular atrophy in gene transfer therapy, and there are other studies looking at gene transfer therapy that we'll talk about as we move forward. The other genetic approach for gene therapy is silencing a gene that is causing bad stuff, meaning a gain of function, a toxic gain of function. And this is usually for dominant disease. Now, I mentioned dominant here, I mentioned recessive there, but just for those that are not genetically sophisticated, dominant means that a one copy of the gene that's defective is enough to cause disease in a person. Whereas recessive means two copies of the gene, one from mom, one from dad, are required to cause a disease. So SMA is recessive, whereas here we're talking about a disease called hereditary transthyretin amyloidosis. It's a form of neuropathy that's genetically transmitted, and there are RNA interference technology and small interfering RNA that are therapies available on the market to treat this disease, transthyretin amyloidosis, with gene silencing. And another way of doing gene therapy is by using this, what's called CAR-T therapy. CAR-T therapy has evolved in the treatment of cancer, but now the idea is, can you selectively target an undesired cell? For example, if you have an immune disease like Myasthenia Gravis, can you target the bad cells, the bad players, and eliminate them with chimeric antigen receptor T cell type of therapy? And of course, all of us have heard about gene editing, and we have all these kind of mixed feelings about it. It's kind of like engineering the gene. And what CRISPR stands for, or CRISPR gene editing stands for, is this long word, clustered, regularly interspaced, short palindromic repeats, easier to say, CRISPR. And typically this is for dominant diseases that are causing toxic gain of function. You take out that gene and then you kind of cause then a recovery from the disease. This is a concept. It's not yet applied in a clinical level. There's no drug that's approved or intervention that's approved in neuromuscular disease, but it's an area of intense interest as a form of gene therapy. Now, with this introduction about what kind of research there is and then the types of gene therapies, I wanted to move on to talk to you about a rare disease. Now, when I say rare disease, and then all the diseases I'll talk to you about are rare diseases, there is a definition for that by the NIH. And the NIH defines a rare disease as a disease that affects less than 200,000 people in the US. So it turns out that when it comes to Lou Gehrig's disease, or ALS, there's about roughly somewhere around that number. It could be closer to 15,000, 25,000, but it's not 60 or 100,000 people in the US. There's slightly more men that have it than women. It can occur at any age, basically. But the peak is somewhere around age 60. And some of you may know about this, about June 2 coming up here very shortly is a special day. Um, does anybody know why? Maybe you want to jot down your answer in the chat box. See if someone knows about this already. I see there's a chat box answer here. Let's see. Am I able to see that? The chat. Or maybe I can't, but
Someone
maybe,
said, someone said Lou Gehrig.
exactly. So the Major League Baseball has an ALS Awareness Day. And this is kind of the link to the announcement by the MLB about June 2 being the Awareness Day for ALS. So this is a very special day. In case you didn't know about it, there's a need to really increase the knowledge base. I know we've had the Ice Bucket Challenge a few years back, but we still need to increase the general public awareness about the dire situation with ALS. So I'm preaching here probably to the choir. Some of you probably have ALS, many of you don't. So please indulge me for those that already have it and already know about the disease. And but this is a disease that causes you to be weak. It spares your brain. It has a typical way of doing it, and it starts with the hand, or it starts in the foot, or it starts with swallowing. That's typically a third of the time it'll start in the arm, a third in the leg, a third of the time it'll start with the swallowing and speech. There are some other rare areas where it can start in breathing or the neck. Um, so what it usually does, it goes from, if it starts in the hand, it goes from one hand to the other, then it can go up to swallowing. Or if it starts in the foot, it goes from one foot to the other, then goes to the hand on the same side as the foot where it started. That's typically how it progresses. And it's a clinical diagnosis. And we know that ALS affects what we call the upper motor neuron, which are those cells coming from your brain down to your spinal cord. And then it affects also the lower motor neurons, which are those anterior horn cells that go directly to your muscles. So the damage is here, but the damage is also there. So this is from a book I edited with Dr. Barron a few years ago. But we know that ALS is overall a bad disease. However, we know that 10% of the patients with ALS live 10% or longer. We know that it takes time to diagnose ALS. We know that there are some risk factors for ALS, but they're not fully known or understood. There's speculation about what causes it. And this is from a paper in Lancet Neurology, a cartoon showing you what kind of things were invoked as a possible cause to trigger ALS development. Because if we know what triggers it, then we can target the treatment directly at the cause of the disease. We think that all of these items here listed contribute to the disease, but we don't know which one and at which stage of ALS, early, mid, late stage of ALS, how they operate at each of those stages. We don't fully understand that. We know that glutamate is a problem. It's too much glutamate, too much excitation of the cells, leading to cell destruction. Free radicals also are destructive to cells. That the mitochondria are small organelles in your cells that generate energy, that drives your cell energy, that there's a dysfunction there, that there's accumulation of neurofilaments, that there is accumulation of certain proteins that are misfolded, like TDP43 and P62. And we know that in the end stage of the disease, or as a disease progresses, that these astrocytes are part of the issue, part of the problem. So there's an interest in neuroinflammation, which is the final common pathway for cell destruction, whether impacting that can be beneficial in people with ALS. So multiple strategies, but no definitive one common pathway that really is a best target of all. Now, we think of ALS as primarily what's called sporadic disease, meaning sporadic means there's no genetic determinant or no gene change in most people with ALS. Turns out 90 to 95% of the patients don't have a genetic disorder, whereas about up to 10% are familial. That's what FALS stands for, familial ALS. AD stands for autosomal dominant, meaning one gene copy being enough to cause disease. Now, when you look at those that are familial, 20% of them are caused because of an SOD1 mutation, and about 50% of the familial cases are caused by this mutation in something called C9orf. It turns out, however, if you look at cases that are called sporadic, you can find that C9orf change in up to 10% of people who are called sporadic. And there are reasons to look for that, and like if there's frontotemporal dementia or a younger age of onset or family history, of course, that's a giveaway. But in sporadic cases on the surface, these would be generally reason to look for it. There are other genes, they're not that common, and it's usually good to do gene testing in patients with ALS when we don't think it's sporadic, because most cases are sporadic. And this occurs if someone has a parent or a cousin or someone in the family that had a history of ALS, or in if there's what's called frontotemporal dementia, which is one of the associations. Now, frontotemporal dementia doesn't mean that people are forgetful. It's a disorder where the organizational or sequencing skills of patients and executive function of patients are interrupted, disrupted. It's not the same as Alzheimer's disease. So patients with ALS do not get Alzheimer's disease. So how do we care currently for people with ALS? Before we talk about research, I think that we know it's extremely important to have multidisciplinary, patient-focused care. And so this is the ALS patient in the center of all of this. And we have the caregivers, whether we have nurses, family, friend, community resources, palliative care if needed, physical therapy, occupational therapy, respiratory therapy, speech language pathology, dietitian, social worker, the research team, and of course, your ALS clinic doctor. So going to the multidisciplinary clinic for ALS care is extremely important. And why is that? Survival time is increased in people with ALS by attending multidisciplinary clinics. And your quality of life, how you feel during the course of the ALS is improved. Your quality is better. Rather than running from one doctor or one clinician to the other from week to week, you go to one clinic visit every 3-4 months, a one-stop shop where you get all your needs met that one day. It's a long day or long morning, but after that, you can live with ALS and not have to worry about going from one doctor visit or nurse visit to the other. So what's the current treatment approach for ALS in addition to multidisciplinary clinic? It has to do with the R3 approach, riluzole or riluzole with Radicava, also known as edaravone. That's a new antioxidant medication that relieves oxidative stress that's been FDA approved in the US for the treatment of ALS. Or it could be R3. You add to those, then research. And let's talk about research a little bit when it comes to ALS. But if I talked all about research only in ALS, I would really be missing important points. I thought it's critically important for all of you to know that we can do a lot right now for ALS. This is clearly there needs to be more, and that's why we're doing research, but it's important not to miss the picture of what can be done at this point in time to help people live better with ALS and their families. So as far as the ongoing key trials that are currently in ALS, this is a summary slide of all different studies that are currently most promising in ALS. Some of you have heard about this ongoing trial out of Mass General and the NEALS, Northeast ALS Group, looking at what's called platform study design. This is testing several different drugs against one placebo group in ALS. You know, one of the issues with clinical trials sometimes is that in the conduct of a study, in a phase 3 study, which is the higher level study, then there is okay, I'm back on. Can you hear me okay?
Yes, we can.
Thank you. I had to take care of a message that popped up on the screen. Thank you. But in doing phase 3 studies, typically the design in ALS is you have a placebo group compared to the medication, and there's 50% of the patients go in one group, 50% go in the other. The idea behind the platform trial is that there will be one placebo group, but 3, 4, 5, 6 groups of people in taking different medications compared to that same one placebo group. The other development is this combination therapy of phenylbutyric acid and TUDCA, where there's been a buzz, a promise about the slowing the rate of decline of ALS, and there's discussion with the FDA about whether this data is within the FDA level of confidence to allow for this to be approved for the treatment of ALS. But I know many patients are taking it over the counters. Phenylbutyrate is not inexpensive. Of course, that's one of the issues. TUDCA is also available over the counter. But we're waiting eagerly to hear from the FDA. People get excited also about stem cell studies. This study, I think, is about finished enrollment. We should have results available soon. I'm not aware of results being published. There's something called antisense therapy, which is one form of gene therapy in those ALS patients who have familial ALS of the SOD1 type. There's also antisense therapy that's being designed for the C9orf subtype, which is the more common subtype. So there's a lot of things going on in ALS research, and this is just a brief snippet of what's going on out there in ALS. I'm going to switch gears and talk about myasthenia gravis. Myasthenia gravis is a disease that causes weakness that varies from time to time in the arms and legs, double vision, droopy eyelids, trouble swallowing. People typically are better in the morning, worse in the afternoon. That's what myasthenia gravis is. The cause for it is an antibody that targets where the nerve and the muscle come together. It's called the neuromuscular junction. These are the currently available treatments for myasthenia gravis: pyridostigmine, corticosteroids, thymectomy, azathioprine, cyclosporine. So there are a lot of different treatments, but recently this drug, eculizumab, which has been a game changer for myasthenia gravis patients, has come on the market. And it's a complement inhibitor, but it's important, and that's why I put it here, to remember that since this drug increases dramatically the risk of meningococcal meningitis, the patient should be immunized to the meningococcus type A and meningococcus type B before they start eculizumab. But there's excitement about something called FCRN blockers, and there are other therapies listed here, and that's why research is very important. So I'm going to come back to some of the research studies going on in the field of myasthenia gravis. This is from the Thymectomy Study. We know now that in people with myasthenia gravis, removing the thymus improves myasthenia gravis and reduces or allows you to reduce the dose of prednisone to a lower level compared to not having the thymectomy. We also know it is safer than not doing the thymectomy in people with myasthenia gravis that are antibody positive to the acetylcholine receptor antibody. And when you look at the gender effect, there's slight differences when it comes to men as improving myasthenia gravis, but clearly it seems to work very well. So this is one of those, as I indicated in the prior slide, one of the first line therapies for people with myasthenia gravis, especially those that are acetylcholine receptor antibody binding titer positive. The next study, and that's the game changer that's currently on the market, is eculizumab. Eculizumab is a complement inhibitor. This is from the study that showed that if you give patients eculizumab, there is this blue line that's eculizumab, a complement inhibitor of decrease, which is improvement in something called the MGADL score, and it gets sustained over time. So that to the point where the FDA approved this drug. Now, the MGADL is a 24-point scale. 24 is the worst MGADL one can have. This is someone who's paralyzed, intubated in the intensive care unit. 0 is the best score. There are 8 items that talk about swallowing, speech, respirations, arm function, leg function, and other metrics. And each is scored from 0 to 3. So you can imagine that a 2-point drop in the MGADL or more is pretty significant in the way of helping people with myasthenia gravis. There are other studies that are currently, the results of which are not yet in the public domain. And these are studies of IVIG that are run by a company called Griffins. And they've ran 3 different studies, one of them to see if the IVIG can help MG. The other one is whether IVIG can help patients get to be on a lower dose of prednisone. And the third study is whether in someone who's worsening, who has exacerbation of myasthenia gravis, if IVIG helps them. So we look eagerly to those results. They're not published yet. I've reported the results of my study, which was an open label study, phase 2, multi-center, where patients who were, as part of their treatment for their myasthenia gravis, already receiving IVIG, I put patients on IVIG dosing 3 times before they went on to get the IG, the same immunoglobulin, but instead of in the vein, which is IVIG, instead of that, they got it under the skin, sub-Q IG. And what we're able to find is that when I put the patients on a 1 to 1.2 ratio, meaning they got to 20% more of the IV dose in under the skin, most patients remain stable. Another area of research is that what's called FCRN blockers. So let's talk about this neonatal FC receptor, which is abbreviated FCRN. What it does, it binds the FC fragment of the immunoglobulin. You can see here, for example, those immunoglobulin going in the cell, and then you have the FCRN that binds the immunoglobulins. When they go inside these endosomes, which are acidic, at that pH, what it allows is the recycling of the immunoglobulin outside the cells. However, if you block this FCRN, then the immunoglobulin stays in the cell and is degraded by the lysosome. It's kind of a cool idea where you can deplete the gamma globulins by enabling them not to leave the cells. They cannot leave the cells anymore. And it turns out that there is this phenomenon of immunoglobulin going in and outside the cells. And this is a method to reduce the level of immunoglobulin. It's similar to what we do with plasma exchange. Now, plasma exchange, we remove not only the gamma globulins, there's cytokines, chemokines, and other mediators, but at least we do remove the gamma globulin, and so this overlaps a little bit with what we do with plasma exchange. And there are several different companies. There are different stages of development of drugs in myasthenia gravis, including Argenex, Momenta, and UCB Biopharma. The other interesting approach would be that Cartier approach in myasthenia gravis, targeting specific bad cells as far as that are part of the process of generation of the antibody that is the problem in myasthenia gravis, and knocking out those cells such that the antibody is gone and therefore the MG may get better. The FDA approval of this type of approach is in B-cell leukemia, in lymphoma, and non-Hodgkin's lymphoma. So there's a study which is called DCART-08, which is listed publicly on clinicaltrials.gov, where they're testing T cells that express this chimeric antigen that targets the B-cell maturation antigen. The B cells are those precursor cells to what's called plasma cells. And plasma cells, what they do is produce antibodies. So if you knock down B cells, maturation antigen, and prevent plasma cells, you're knocking down basically the antibody production. So there's a study of that sort, and that is currently ongoing. I'm going to switch gears and talk about myositis. And this is from the criteria for diagnosis, but I'm not going to walk you line by line through those criteria, but I thought I wanted to show you what I teach to medical students, residents, fellows, and faculty at different conferences. But generally speaking, someone with progressive muscle weakness, high muscle enzyme, abnormal EMG, a muscle biopsy with inflammation has myositis. Now, dermatomyositis on the muscle biopsy shows what we call perifascicular atrophy, meaning if this is a cross-section of a muscle biopsy, this is the center of the muscle biopsy, this is the outside of the muscle biopsy, and you can appreciate that those fibers are a bit smaller compared to these fibers. That has a name for it, perifascicular atrophy. There's also inflammation you can see here in an area called the perimysium around blood vessels. Perimysium is one of the connective tissue layers, and you can still see the atrophy here of the perifascicular area. Now, in addition to the pathology in dermatomyositis, you can see these typical rashes. They're called Gottron's rash, Gottron's sign, Gottron's papules, and you can see them on the knuckles, you can see them on the joints of the fingers, you can see them on the knees, on the elbows, the extensor surfaces, and this violaceous scaly rash or the heliotrope rash are also typical of dermatomyositis. We know that 70% of patients with dermatomyositis have one or another antibody, and they convey different types of things. I'm not going to go through all of this. That's probably too much information. But in children, for example, this antibody indicates that they're going to have a lot of skin disease. This antibody called MDA5 shows something like skin ulceration. NXP2 is not the only association, I'll show in the next slide, but they can have quite a bit of calcium deposits called calcinosis and swelling in the legs. SAE can have a lot of skin presentation before the muscle gets weak. But we know one of the associations with dermatomyositis is cancer. About up to 20% of patients with dermatomyositis will have cancer. And this is typically within the first few years of the onset of the disease. It could be already there for a couple of years, but we have to be watchful for a few years from the time of diagnosis. It turns out that those two antibodies represent the majority of cancer cases. So how do we treat currently the myositis, dermatomyositis, for example? Prednisone, methotrexate, azathioprine. These are immunosuppressive drugs, IVIG. And recently, Octapharma concluded a positive study of IVIG in dermatomyositis, and there are others listed here. But let's talk about research, because that's our goal here. And these are some of the clinical trials that are going on in dermatomyositis, and I'm focusing on those clinical trials that have occurred and we're involved in. There's a study of abatacept by Bristol Myers Squibb that is looking at polymyositis and dermatomyositis. It just concluded enrollment. We don't know the results of it yet. A study that's currently actively enrolling is a study by Kezar. This is to modulate the immunoproteasome. And this is an inhibitor of the immunoproteasome. And so it's a novel mechanism of action that's being tested in dermato and polymyositis. Ra Pharma was acquired by UCB, and they tested a complement inhibitor in patients with statin damaging to the muscles. It's called HMGCR, antibody type of myositis, or SRP type of myositis. Unfortunately, we just learned recently that the drug did not seem to work in those patients. Corbus is another company that is engaged in testing a drug called linabasum in dermatomyositis. And this is interesting because this drug doesn't give you a high, but it's a cannabinoid receptor agonist. But there are multiple receptors for the cannabinoid. This is cannabinoid 2 receptor agonist. So it does modulate your immunity, and we're hopeful, but the study is ongoing right now. We don't know the results of it. So a lot of studies going on. I showed you just a brief snippet of what trials are going on, and we already have a success this year in the sense of the Octapharma study showing that IVIG does work. And the hope is that at some point in time that the FDA will approve the drug for dermatomyositis. This way we will have less hurdles for our patients with dermatomyositis when we want to treat them with IVIG to help them live better with the disease. So to close the loop, I'm going to talk about the fourth disease state, Pompe disease. And since this is an adult conference, I'm going to focus on late onset Pompe disease. And this is 2/3 of all Pompe cases. And it's characterized by some leftover enzyme activity in the skin. You can measure it in the skin, you can measure it in the blood. And typically it's up to 40% leftover activity. It's not 100%. 100% is normal. It's 40% from the lower limit of the acceptable level of activity up to 1%. If 0%, this is when it's the infantile form, IOPD. We're not talking about that today. So they can present at any time. That's the key thing. But there's no heart involvement in the first year is what distinguishes late onset Pompe disease. So if someone can present at 9 months without heart involvement, that's still called late onset Pompe disease. The infancy onset, they have a lot of other issues, and they're listed here. Whereas in childhood, there could be some delayed milestones. There's left ventricular hypertrophy, which is enlargement of the left chamber of the heart, some arrhythmias or irregular heartbeats in the later onset group. And not everybody is the same. Everybody is different. We have now the screening test for neonates, neonatal screening test in different parts of the country. Missouri and others have newborn screening tests for Pompe. So one of the things we're dealing with nowadays is you find someone who tests positive, a baby that tests positive on newborn, but what do you do with that information? If they're not weak, they're moving their arms okay, there's not obvious disease, how do you follow them over time? How do you initiate the treatment at the right timing, knowing that they're not the IOPD category? But before we get to that, I think it used to be in the past that we did muscle biopsies to show that there's deposition of a sugar called glycogen, to show that there's disruption of the muscle fibers, and there's these what's called acid phosphatase positive vacuoles. But now we can do gene testing on patients, and we can capture most cases that way. The enzyme activity is also helpful. Currently, there's enzyme replacement therapy. Lumizyme is FDA-approved enzyme replacement therapy for Pompe disease. But let's talk about research in Pompe disease. And again, I did not know Amicus is one of the sponsors. And as you can see here from this slide, I'm not favoring one drug over the other. I'm going to just show you what's going on there in Pompe in the way of research. I think Amicus completed the study of chaperone to increase how long the enzyme replacement therapy stays in your body, because some of you who have Pompe know this is given every other week. So, and typically the course of response of patients with Pompe, once you start giving it in the late onset group, once you start giving it every other week, is that in the first 2-3 years, people improve, they peak and plateau, and then they start declining after 4 or 5 years. And we never could understood why that kind of curve occurs. So the idea behind the chaperone to protect the enzyme replacement from degradation, that's what the chaperone do, they're protective molecules. Can you increase the length of survival of that enzyme replacement therapy in the bloodstream, keeping it every other week in such a way that you don't have that decline later on? And that's what the Amicus program is about. They're using their own enzyme replacement therapy too. Now, the eval glucosidase alpha is a different ERT. This is Sanofi that's developing that. This is a drug that's currently undergoing, probably will be undergoing later on this year, FDA review about approval possibility. And this is a different enzyme replacement. It's called eval glucosidase alpha instead of L-glucosidase alpha. This is modified so that the enzyme replacement therapy can better penetrate the muscle fibers. The muscle cells have on them something called mannose 6-phosphate receptor. This is that receptor. For the enzyme replacement that you give in the vein to go into the muscle fiber, it has to go through this to engage with the mannose 6-phosphate receptor. So this reformulation of the enzyme better targets the mannose 6-phosphate receptor. But of course, all of us get excited about gene therapies. And here, of course, there are several programs, and I'm mentioning some of the more advanced programs, not the ones that are in the preclinical or earlier phases of development. But Actis has a program, Odentis and Spark has a program, and I listed them here alphabetically, not in any order of preference. So we need people to participate in research studies because gene therapy is a possibility for people with Pompe. And so I encourage everyone with Pompe to consider that as an option. A lot of the requirements for these studies is that you have to be on stable dose of enzyme replacement therapy for at least 2 years. So if you're not on treatment, you may not qualify, and there are other inclusion-exclusion criteria that are listed. But it's really exciting to know that there's an interest as well as a potential for getting therapies that will be game changers for treating Pompe disease. So this is my conclusion slide. Right now, for when it comes to neuromuscular MDA type diseases, dystrophies, and related diseases, we can diagnose diseases quicker. We have better tests. We have more confidence in the diagnosis. We can do a lot right now. Through multidisciplinary clinics, we can help patients quite a bit, improve quality of life. We can give symptom treatment, but also, as you noticed, enzyme replacement therapy is specific therapy. Complement inhibition for myasthenia gravis is specific therapy. So there are specific therapies. We have more clinical trials, newer designs. There's more access to or expansion of gene therapy possibilities. And I'm hopeful that as we talk and continue to talk about disease-specific areas later in the year with SMA and other diseases, that you'll hear more about specific advances that bring hope and relief to people suffering from neuromuscular disease. So with that, I will stop here, and we have a few minutes for Q&A. Thank you for your attention.
Yes, we do. Thank you very much. We do have a couple questions, so I just want to go ahead and start with what we've got. This person says, I have myomyopathy and second carnitine deficiency. I have been put on NADH, which has decreased my fatigue and increased my endurance. Yes, I am on carnitine and a mito cocktail. My question is more around the hormone levels that tells your body you need ATP, especially using fat. I don't see any clinical trials, but as you very well stated, there's a bench work before. Do you know of anything that's going on in that area? And that was from Crystal.
This is a very good question, and then illustrates how there's a large variety of diseases. So the question is about a form of mitochondrial disease where currently there's not a good treatment at all for this condition, and we give antioxidants, vitamin A, vitamin C, and other antioxidants, vitamin E and other antioxidants as part of a cocktail, yet we're not sure how good these cocktails work over time. In this case scenario, I can think of two centers in the US that would be the expert centers in mitochondrial disease that I would want to get their opinion on whether there's an answer to this question. And clearly there's no clinical trial. I know Dr. Haller, Haller, in Dallas at UT Southwestern, is one of the international experts in mitochondrial disease, and Dr. Michio Hirano at Columbia would be the other expert. So unfortunately, I do not have an answer to your question, but I can direct you as to who would be my choice of experts to visit with regarding that question. I'm located in Kansas, so typically when patients with mitochondrial disease present themselves, I always enjoy the fact that Dr. Haller is just a long drive away from Kansas City, but an excellent resource for my patients.
Thank you for that. This is a question surrounding the COVID vaccine. They have CMT1A, and they're wondering if they should get the COVID vaccine. They say, I had Guillain-Barré and double Bell's palsy after the flu shot, and it was devastating to their mobility. Is the COVID vaccine something that you recommend for folks with CMT1A?
Well, the Guillain-Barré CIDP...
Sorry.
I'm sorry. Did you want to say something else?
No, I just mispronounced that. I apologize.
That's fine. No, you're fine. It's a French... Actually, Guillain was a French army physician in 1914, as well as Barré. They worked together to describe that entity. So it's hard to pronounce those French names. It's well understood. But let's talk about CMT1A in isolation. Then I'll come back and talk about Guillain-Barré syndrome, because that's a multifaceted question. But particular to CMT1A, and most of the disease we're dealing with here, I don't know if there are exceptions, I have recommended quite uniformly for my patients to get immunized against COVID. I've seen little reasons to, the key questions that come up, well, I've had COVID 90 days ago or 30 days ago, should I get the vaccine? And I tell them, well, you know what? You may have more of a reaction because you have some immunity in your bloodstream right now, but that's the only reason to be pausing for a concern. You may have more muscle aches, more headaches with the early immunization. You could do it. I usually encourage them to do it earlier. The other question comes about, well, I'm on immunosuppressed type drugs. I'm taking prednisone or other drugs. Should I take it? And the answer is all the more reason to take it. Your immune response to it may not be as good and vibrant as someone who is not taking prednisone or other immunosuppressants, but some protection is better than no protection. Now, in CMT1A itself as a neuropathy, I don't see a contraindication to take the COVID vaccine. The GBS CIDP Foundation, when it came to Guillain-Barré syndrome, which is called GBS, also is easier to pronounce it that way, came up with a statement saying that all patients should get the COVID vaccine. There's no good data out there to tell us about if you've had GBS in within the 6-week time period from a flu shot about what happens with the next flu shot. There's only one study from the UK that was a population-based study where they looked at all patients with GBS and sent them questionnaires. Some responded. And the bottom line is the likelihood of worsening is small, even if you've had a GBS associated with a flu shot. If you got the next flu shot, the likelihood is, or another vaccine, the likelihood is low for worsening. And most of the time, it's a minor worsening. So I advise my patients with GBS, especially if after a vaccine, especially if they've been like year or more away from their episode of GBS, generally speaking, to get the vaccine. For that specific question, I encourage the person that asked to consult with their physicians because there's sometimes modifiers to that answer. But generally, the answer is yes.
Okay, thank you. Is there any new research for Nemaline myopathy?
Nemaline myopathy is one of there's a sporadic late onset Nemaline myopathy, and there's a Nemaline myopathy that's genetic. And one of the things I was going to try to do, but I deleted in the interest of time, is to teach people how to go to clinicaltrials.gov. It's a website. And then you put in the name of the disease, and all of a sudden, all the research studies that are recruiting become available to you and physically visible to you for you to see them. And so maybe, can you screen share that, Nicole, from your end? I'm going to stop sharing here.
I just typed it in the chat for everyone. It's clinicaltrials.gov.
And then you put in Nemaline myopathy, and there you'll see, and on the left side of the screen, once you Nemaline myopathy search comes up, there may be 10, 20 studies, but the way you filter it is to click on recruiting because you are interested in what are the studies that are recruiting. And once you do that, then you'll find out, and actually, I may have prepared the slide, but never included it. Let me see if I cannot find it. And that would be probably a nice way to illustrate the point. My slides are opening. It'll take a second here. So I did it for Pompe disease. Let's see, screen share. And this is the one. Do you see my screen now?
I do.
Okay. So this is how to find studies. Okay. So basically, you can consult with your NDA care team. You go to clinicaltrials.gov. You plug in the name of the disease right here. Okay? You put Nemaline myopathy. Then next thing that you do, in this case, I put Pompe disease. Okay? Once I put Pompe disease, it tells me there's 48 studies. And you can see here, not yet recruiting, enrolling, all that by invitation. You want to click on recruiting because you want to know how to get into an individual study that may fit your profile. So once you do, and I put recruiting for Pompe, it came up with 12 studies. See that? And I showed you some of those studies, and we talked about some of them. Gene transfer therapy and other therapies are listed here. This is the Amicus study. So you can see all the studies that are available. And once you click on them, then you get more information. I don't think the next slide is about that. So
thank you for sharing that. That's wonderful.
Yeah, thank you. But thank you for asking the question. I prepared specifically for this meeting, but I decided not to include it because of the interest of time. But thanks for the question. That helps me use it.
Thank you for that. Are there any medications for FSHD?
Right now, I think Dr. Statland, my colleague here at University of Kansas Medical Center, is one of, if not the ultimate, FSHD expert, and he's working on different studies and outcome measures for the development of therapies for FSHD. But there's a large natural history studies that includes the US, Europe, that he's leading. So if you're interested in that, we can connect you with Dr. Statlin and his team. There are multiple sites around that. And this way, you'll be very close to the next research studies when the research study when it starts.
Perfect. Okay. This person has CMT1A and they're using a non-invasive Trilogy respirator, but her breathing has become shallow and has increased tiredness and decreased energy. Is there anything they can do to improve breathing?
Yeah, this is very specific pulmonary question. Typically, when patients ask me that, I get the help from our pulmonary specialist and critical care and figure out what would be the ways to turn it. Also talking with the respiratory therapist, that is in the clinic about what can be done to improve that would be my suggestion.
Okay. Have you had anything that has come about with limb-girdle patients being misdiagnosed with polymyositis? Have you encountered that at all?
Yeah, it does happen, actually. I was looking at a record of a patient recently who had a biopsy elsewhere.
Uh-huh.
In the biopsy report, it was mentioned this patient has myositis. This is a couple of years ago.
Okay.
But this patient has polymyositis. They have to be on steroids and all that. But when you see the patient, you realize that even though the biopsy picture can be fooling you, but the clinical picture of slow progression over time, over 20, 30, 40 years of weakness that goes back to even high school days, that kind of gets to be a big clue. So yes, that's been reported. Some of the muscular dystrophies like dysferlin and can cause inflammation in the muscle. They can be confused with polymyositis. Also, anoctamin 5 defect, which is another form of autosomal recessive muscular dystrophy, and others can have a similar picture that overlaps with polymyositis. And so, yeah, it's understandable that at times there are misdiagnoses. One of the other misdiagnoses with polymyositis is inclusion body myositis, but that's not a genetic disease. So yeah, there are opportunities for better diagnosis. I tell patients, if you're seeing a local neurologist that specializes in nerve and muscle disease and neuromuscular specialist, you're in good hands. And if you're in a doctor that's very well versed in neuromuscular disease, you're in good hands. But if you get the sense that, they see mostly patients that are not in that type of specialty, you may want to ask for more specialized care.
That's a
And the
really good
MDA
point.
has a lot of good resources.
Yeah, that's a good point. Is Pompe disease considered a metabolic disorder?
It is a metabolic disorder in a way. It's a
Okay.
lysosomal storage disorder. And it's those lysosomes that kind of degrade things. There's a storage of glycogen material inside the cells because of the inability to process it.
Okay. All right. Um, can you tell us if there's any information or any new research that's gone on for the ocular pharyngeal?
So oculopharyngeal muscular dystrophy, that one I would say, let's go searching on clinicaltrials.gov. Let's find
Okay,
out what new
perfect.
research is out there. I know there was a presentation from an investigator in Israel about an early study drug development program in oculopharyngeal muscular dystrophy that was promising. However, I don't know if that was carried on to the next phase of development. Dr. Argov is his name.
Okay. And just due to time, I wanted to follow, I think this is a great way to end this segment. What advances in recent neuromuscular research have you found most exciting, and what future advances are you most looking forward to as research and clinical trials in this field progress?
Wow, this is a very nice question to
Isn't it
really
great to
end
end with?
with. Yes, I think there's a lot of reason to be excited about the future of care of people with neuromuscular disease. Let me tell you, having been doing this for the last 3 decades, there was a really long time stretch where neuromuscular diseases and MDA diseases, from a research standpoint, were forgotten by the pharma companies. Not totally, but for the most part, very, very little in the way of number and volume of studies. And from investigator standpoint, there was less throughput in the way of research. But right now, everybody is on board. All
Mhm.
hands are on deck. So I'm very excited about different study designs, this platform design, particularly for ALS, the potential for gene therapies at different levels. And I showed you the different types of gene therapies. Now they are available to us. This was kind of a big dream, but we need more of those. And I think we'll see more of those in the next few years become available to help our patients with muscle nerve disease and ALS and neuromuscular junction disorders. So thank you for this question.
Yeah, perfect. Well, thank you very much for your time on Saturday. We definitely appreciate you being here. And thank you for answering our questions. I know we had some few more questions. We've tried to answer all our questions, but we do have our next speaker coming up on nutrition. So thank you very much for being with us today.
Thank you very much. Have a great day.